Hello everyone and welcome to this podcast for epigenetics. In this podcast I'm going to finish the discussion of the somatic cell nuclear transfer. If you remember, last time I had cut that short because of some technical problems. So I'm going to finish that up first, which will actually take the majority of the podcast. And then I'm going to spend a little time talking about reprogramming by cell fusion. So let's go ahead and get back to the somatic cell nuclear transfer experiments. Now remember, adding that nucleus into that oocyte is not something that naturally occurs. So that oocyte, it tries to demethylate it, and it does a pretty good job, but it's not perfect. So what I want to say here is, sometimes even that serial nuclear transfer or the extra time with NPF still isn't enough time to completely reprogram the epigenome. In fact, I should probably use a stronger word here than sometimes. Usually, it's not enough to completely reprogram the epigenome. Sometimes it reprograms it enough to get that viable progeny. And sometimes it doesn't, and sometimes you don't get the viable progeny. So if we think about it, when we take this oocyte and we place that somatic nucleus inside of it, a few things have to happen in order for us to eventually go through embryonic development and get a cloned progeny. And it must stop expressing genes specific for epithelial function. Now, I say epithelial because all along we've been talking about taking the diploid cell from an epithelial cell. But if you took it from a liver cell, if you took it from a heart cell, wherever you took it from, you need to turn off those genes that make it an epithelium and makes it a heart cell, makes it a liver cell. You have to turn those genes off. This DNA here that has been introduced must become responsive to instructions provided by the oocyte cytoplasm to turn on developmental genes like OCT4. Remember, OCT4 is going to be important to be turned on so that when we begin to make pluripotent stem cells, that it can maintain those pluripotent stem cells. In the epithelial cell where this DNA originally came from, this gene would be turned off. It does need to be turned on now, but what this point is saying here is the factors in this cytoplasm, this DNA has to be able to respond to it, so we can turn on specific genes. This last one here is maybe the toughest one. The heritable memory of the donor DNA must be erased. Those histones that are attached to certain genes, certain areas of the chromosome, have to be detached, so they're loosely associated with the chromatin. So like H3K9 methylation, one might predict should be removed to make it go from heterochromatin more to euchromatin. Because remember, we need the DNA here to be mostly euchromatic so that it can have that potential to become any particular cell. So these three things have to occur. Now, not surprisingly, there's a lot that has to happen. And not all of this occurs, or at least not all of it occurs to 100%. We know when we look at these embryonic cells found here in the middle here, there's at least 4% misexpressed genes. I guess that's not horrible. 96% of them are doing what they're supposed to do, but about 4% are misexpressed. But I suspect the reason that we don't get 100% cloned progeny is because of this imperfect resetting of the genome. We see more misexpressed genes in the trophoblast cells, those that will go on to become the placenta. And we think that might be because those cells are set much sooner than those of the inner mass cells. So they might not have had enough time to reset. And so we do see a little bit more misexpressed genes in the placental cells, the trophoblast cells that then go on to become placental cells. Now, an interesting thing here, and I hate to just put it on the side here because it's a really important point, so make a note of it, is that of these 4% or so misexpressed genes, imprinted genes are overrepresented. meaning that of the genes that are misexpressed, it's usually imprinted genes that are misexpressed. Now remember, as we set the genomes and as we go through these early embryonic stages, we know that the DNA is demethylated, but the imprinted genes are usually not. Those are usually preserved until you start making the germline. So it's very unusual that the imprinted genes somehow are more likely to get misexpressed compared to other genes, non-imprinted genes. So the end result is usually imprinted genes, we only see half of the genes expressed because either mom or dads are imprinted. But now what we're seeing with some of these imprinted genes that we have twice the expression. So let's spend a little bit more time talking about these imprinted genes and why they are more likely to be misexpressed during SCNT development. Remember, imprinted genes often can respond to environmental changes or stresses. Sometimes we can get misexpression of these, or we can get changes in expressions of them. So it's possible adding the nucleus in there from a somatic cell is enough to respond in this environmental way that allows some of these imprinted genes to be misexpressed. We haven't said this before, but it's a good point to make here. When artificial reproductive technologies, ART, are used, so like in vitro fertilization, there is often an increase in the probability of having an imprinting genetic defect. We're more likely to see the misexpression of imprinted genes when things like in vitro fertilization or other kinds of artificial reproductive technologies are used. So I suspect it wasn't too overly surprising that they made this observation with the somatic cell nuclear transfer. So a question that might pop in your mind, and the book proposes too, is why does this work at all? It almost seems shocking that this could actually work. And the reason that it works probably is that this somatic DNA hijacks a system that's already present. And so let me explain what I mean by that. So let's recall that when the sperm is made, that the DNA in it is highly packed, even more so than thinking about the mitotic nucleus, where it's all heterochromatin. It's packed so tightly that histones aren't involved, so none. Instead, it uses structures called proteamines that wrap tightly around the DNA to condense it. And the only reason this works, these proteamines, is because the DNA is overmethylated. So we know that when the sperm fertilizes the egg, that the proteamines are rapidly removed, as are the DNA methylation sites. DNA is then repackaged using the maternal histones. So the DNA comes in here, the proteamines and the DNA methyl sites are removed, and then the DNA is repackaged using maternal histones. So it's possible that if I were to draw it down here, we take our enucleated, remember this isn't nucleated, it has the oocyte nucleus in it, but we take an enucleated egg and we add a diploid nucleus in it that is heavily methylated because it's been differentiated and there's a lot of heterochromatin placed in there. There's a system already present in the egg to remove those methyl groups. And so perhaps this system, as it removes these methyl groups here, helps reset it in pretty much the same manner that a natural fertilization occurs. However, not at the same rate, because it is much slower. But then it can be repackaged using maternal histones, as we might normally see. This could explain why the longer it's present with the factors in the cytoplasm helps the success of this forming an actual embryo and progeny. So we often think of this DNA here being placed in this oocyte as hijacking the natural system that it already. So how does the oocyte reprogram? That is, how does it get rid of the methylated genome? There are three ways it could do this. It could remove the methyl groups through demethylation enzymes. There's a gene called MBD2. MBD stands for methyl binding domain, that is thought to maybe play a role in this. Another idea is there could be base or nucleotide excision repair. This is a repair mechanism that's usually used to fix DNA mutations, but maybe it's being utilized in this sense to remove the methylated DNA. And the gene that's thought to maybe be involved here is called MBD4. The last one, which has probably the least amount of support for, is that there could be a deamination of the 5-methyl cytosine to produce thymine. This would require a great deal of energy to remove all of them, so that's one reason we think this is problematic. Another reason is you would create the wrong base, and that could have downstream consequences that would not benefit the progeny that survived, if they survived. Okay, a couple things I want to talk about are these two questions. The SCNT demethylates slower. And the next question is, why are SCNT cells, why is it that imprinted genes demethylate faster? Because you get that mis-expression of these imprinted genes. So these two, in my opinion, as I look at these, are kind of counterintuitive to each other. Doesn't make sense that most of the genome demethylates slower, but not the imprinted genes. They demethylate faster. Well, let's consider what happens under a normal fertilization. We have our nucleus here. Two things happen. We have methylases that need to demethylate the DNA, and in that process, we want to make sure we keep this enzyme, DNA methyltransferase, the one that adds methyl groups, we want to keep that out of the nucleus. In this system here, one might imagine we would demethylate the entire genome. But what happens under natural fertilizations, there's this DNA methyltransferase that we haven't talked about called DNMT00. It's an oocyte-specific DNMT, and it will enter the nucleus. And DNMT00 is specific, or more specific, to imprinted genes. So under normal circumstances, methylases come into the nucleus, they demethylate the DNA, but to help protect imprinted genes so they're not demethylated, this DNMT00 is expressed and is in the nucleus, and it helps to maintain the imprinting, the methylation of the appropriate imprinted genes. So this was natural. Now with the SCNT genes, or cells, you have this somatic nucleus here, and usually what happens over here, it obtains under a natural situation, I should have mentioned this before, is it contains lots of H3K9 acetylated histones. In fact, the sperm, when it comes in here, that DNA uses the maternal H3K9 acetylated histones that are in the cytoplasm. What this does is it opens up that DNA, not for transcription, but it opens up that DNA so that methylases can come in here to demethylate the DNA. So over here, that's not happening, or at least not as efficiently. We're not getting as much H3K9 acetylation in the diploid nucleus here. And because of that, it demethylates slower, because the enzyme that would have to facilitate that transfer isn't able to access the DNA as efficiently. Likewise, this DNMT00 is still there, and it's still going into the nucleus, but for the same reason that the methylases that remove the methyl groups don't have access to the DNA, the DNMT00 also doesn't have access to it. And so you're kind of having this battle, at least this is what is hypothesized, having this battle between the DNMT00 trying to keep these methylated and the DNA methylases battling with them to remove them. Both inefficient processes. And because of that, you will get some of those imprinted genes being demethylated, which can lead then to the misexpression of those imprinted genes. Not all of them, again, because it's a battle, and so sometimes the DNMT00 may win out, and you get proper expression of those genes. Okay, the last thing I want to say is just an interesting observation that provides some evidence that the reprogramming is not perfect, and that we have some problems with it. And that is that our gene, OCT4, if you remember, this is the gene that helps maintain the pluripotent state. It is expressed at a lower rate. We don't have as much OCT4 being expressed. Now that's not always a problem, because often we do get a progeny that forms. But because it's lower, that might help explain why these cloning methods don't always work, because you're not able to form that or maintain that pluripotent stem cell state as long. Okay, now let's move on to talking about cell fusion and reprogramming. So in these experiments, they were just curious if they could take a somatic cell, and they usually use a B cell. Not sure why, but they do. They take the somatic cell and they combine it with an embryonic stem cell. So they both would be 2N. And when you combine these two together, what happens is that these two cell membranes will fuse, and you end up with something we call a heterokaryon. Hetero for different, karyon for nucleus. One is 2N from the somatic cell, and the other is 2N from the embryonic stem cell. Now what will happen is these two nuclei will fuse with each other, and we'll end up with that same cell, but now it will contain a nucleus with 2 chromosomes of each from the embryonic stem cell and 2 chromosomes of each from the somatic cell. And it behaves as a pluripotent cell. But keep in mind, it is a tetraploid. That is, it is 4N. Now, as you can imagine, very few survive to this point. Lose a lot in this whole process, but still, surprisingly, some still survive. And more interesting is that the ones that survive, the somatic chromosomes, those that came from the B cell, are reprogrammed to appear as embryonic stem cells. Mainly, they express pluripotent cell maintenance genes like OCT4 and SOX2, as well as other genes important to maintain that pluripotent state. Now you can take these tetraploid stem cells here and you can grow them in culture. And they'll do fine for a while, but eventually they're unstable. And in a very interesting way, they begin to lose half their genomes. But not any half of their genomes, just the somatic cell genomes. So they begin to lose the somatic cell genomes in a very selective way. Then once they are lost, then the cell lines are once again more stable. This suggests that maintaining the pluripotent state is cis acting and less trans. What do I mean by that? Well, let's talk about that for a moment. Now, interestingly, when I say that the somatic cell is reprogrammed, it's reprogrammed in the heterokaryon. There are factors that somehow will enter into this nucleus from the somatic cell and reprogram it to turn on OCT4 and some other genes. And we would call that trans-acting. Something from outside, the immediate chromosome being affected, is acting on that expression. But for some reason, when this heterokaryon, the nuclei fuse, it will maintain some ability to divide, but this is when we start losing the somatic chromosomes. And we believe that is because there is a cis-acting element that acts with the embryonic stem cell chromosomes. What that is, we don't know, but remember, for something to be cis-acting, it has to be on the same chromosome. So it could be something, and I'm not saying this is what's happening, but it certainly is a possibility, I would suspect, is if you have an imprinted gene, and we have these three, say, mRNA coding genes, and how their expression is dependent upon this non-coding RNA. And often if this is expressed, these aren't, and that's how we imprint these genes. This would be considered cis-acting, because this is directly affecting the expression of genes next to it on the same chromosome. So it's possible that in the embryonic stem cells, there is an activation of a non-coding RNA that keeps these genes off. But in the somatic cells, it's the reverse. These genes would be expressed. You could also imagine that in embryonic stem cells that the non-coding RNA is not expressed, and so that these genes would be expressed. But we suspect it's cis-acting, since we specifically lose the somatic cell chromosomes and not the embryonic stem cell chromosomes.